new edition of the Xylella Seminars that is jointly organized by the BCIL projects, the Oban University, the EFSA, the European Food Safety Authority, and CSIC. And so good morning or good afternoon, depending on from where you join us. And then I will give the floor to Evelina Stinsek that will present our two speakers that we have today.
Hello, everyone. Thank you, Juan. Thank you, Anna, and thank you, Blanca, for organizing also this session. Today, finding of Xylella fastidiosa ST1 in almond crops in Apulia via vector surveillance. This is a topic, and we have very special speakers, two speakers, Daniele Cornara and Maria Saponari, who are very well-known scientists from Italy, from Bari. And Maria is working for Italian National Research Council Institute for Sustainable Plant Protection. And Daniela is now presently, correct me if I'm wrong, assistant professor at the University of Bari in general applied entomology, specialist in vectors. I would emphasize Xylella vectors. And if you can share your screen, Daniela, because you will start, it's a pleasure to have you here. Please share your screen and go ahead. And maybe some in-house rules here. I would recommend to close the videos, speakers for the people who are not speaking, who are not the prelegant. And the rest, there will be a moment for the questions. Right, Juan? Do you agree with this?
Yes, they can use the chat to write the questions. That will be maybe respond after the presentation.
Excellent. So over to you, Daniele, now. Let's start.
So can you hear me and see my screen?
Yes. Yes,
okay,
also
cool.
online.
So it's a pleasure. So thanks for inviting me, and let's get started. So most of the people I'm not talking about scientists, when thinking about Xylella fastidiosa, they tend to think about a bipartite system with a plant and a xylem-restricted bacterium that can cause devastating disease as olive quick decline syndrome or Pierce disease or citrus variegated chlorosis, and that actually is considered a global threat. And as we all know, to react to this global threat, one of the actions which are undertaken is Xylella surveillance. So trying to understand where Xylella is in a certain area, if present or not, if present like in a transient way or established, so on and so forth. And this approach actually in Europe is mainly a plant-based approach. So when looking for a certain bacterial genotype, we go to the field, we sample some host plant species, selected host plant species, and then we collect these plants, sample of these plants, we go to the lab and we test these plants for Xylella fastidiosa. But this approach has, obviously as all the approaches, some biases. For example, when we are looking for Xylella fastidiosa and certain plant species, so we go to the field and sample certain host plants. But if another bacterial genotype is present in the area and we are not sampling the host plant of that bacterial genotype can go undetected in the area. Plus, there are other factors of the Xylella pathosystem that make the survey pretty complicated. For example, the absence of specific symptoms caused by Xylella, or the heterogeneous distribution of the bacterium in the plant, or the long incubation period, and so on and so forth. So the question is, how can we improve the efficacy of this Xylella survey system? And one of the way is looking at the third actor of what is really a tripartite system: plant, bacterium, and insect. So what really moved Xylella fastidiosa? So the xylem-feeding insect vectors. So mainly sharpshooters and spittlebuggs, and you can see at the top bottom a picture of His Majesty. So the most epidemiologically relevant vector found in Europe in all the outbreaks so far, which is Phyllanus spumarius. So basically, if we include like Xylella surveillance with an insect-based approach, looking at Xylella inside the vector population, we can overcome some of the biases of the plant-based surveillance. For example, there is no vector species, bacterial genotype specificity. So we don't care about the host plant species, we don't care about incubation, disease, and so on and so forth. We basically collect insect and test the insect for Xylella fastidiosa. But there is a main problem using vector for vector surveillance. And its problem is, as usual, knowledge gap. So compared to the plant-bacterial interaction, so not that much is known about the transmission biology of Xylella fastidiosa by spittle-bugs. So since the first detection in 2013 in Europe of Xylella fastidiosa in Salento in olive, so several scientists carried out transmission trials with spittle-bugs, with Phyllanus spumarius, to understand pivotal factor for the transmission of Xylella fastidiosa, which is the transmission efficiency. And we estimated the transmission efficiency by Phyllanus spumarius of Xylella fastidiosa as 6 to 11% for the olive-to-olive transmission, or around 15 to 20% for the grape-to-grape transmission. So it's a competent vector, it's an efficient vector, but not that efficient as, for example, the blue-green sharpshooter in the transmission of Xylella fastidiosa from grapevine to grapevine. Here we are talking about a transmission efficiency which exceeds 90%. But the main factor for Phyllanus spumarius that make Phyllanus spumarius a pretty scary vector of Xylella fastidiosa and pretty efficient vector of Xylella fastidiosa is another one, and is that NT, which is called in our jargon vector activity, which is the product of the number of individuals that are on a plant per the time they spend on the plant. And the problem here is that we don't know that much of vector activity for Phyllanus spumarius within European agroecosystem, olive orchards as other agroecosystem. We don't know how Phyllanus, for example, relate to the landscape, how Phyllanus detect host plant, what make host plants suitable for Phyllanus, how Phyllanus interact with biotic and abiotic factor within the agroecosystem, and what's, for example, the impact of agronomic management on the vector activity which influence Xylella fastidiosa transmission. So with this in mind, Regione Apulia decided in 2022 to start a project, the project SILVET, which is a 3-year project which has been entrusted to the Vincenzo Verrastro's group at the CM Bari under the scientific supervision of the University of Bari, and in collaboration with other projects which are investigating spittlebug and transmission of Xylella fastidiosa, which are the Covexi and SOS, founded by the Italian Ministry of Agriculture, and the NOSE projects founded by the Italian Ministry of University and Research. So the 3 main goals of this project is trying this vector surveillance approach. So using vectors to try to detect Xylella fastidiosa in Xylella fastidiosa free area, and basically looking at olive orchards, because of back in 2022, we only knew about the presence of the ST53 in Apulian olive orchards. Second, providing farmers with an indication about the correct timing for vector management through ground cover removal and chemical treatments. And third, trying to collect data about the key factors that influence the vector activity and the transmission of Xylella fastidiosa in Apulian olive orchards. So basically, what we do is we selected 90 olive orchards in the Xylella fastidiosa free area, olive orchard with different landscape characteristics, so surrounded by semi-natural habitats, or olive orchards, or grape, other crops like grapes, stone fruits, almonds, so on, in different characteristics in terms of elevation, in terms of climatic characteristics, but also different agronomic management. So in each of these orchards, we conduct every 10 days survey for xylem feeders, starting from the juveniles. So the juveniles are like monitored through the quadrat sampling method, collecting data about their presence, abundance, development, and host and non-host plant, again, to give farmers indication about the most appropriate timing for ground cover management to curb the vector population inside the olive orchard. The adults are instead monitored by SweetNet on the ground cover, olive canopies, and border plants, mainly trees and shrubs, using a non-destructive approach. So we collect the insect, we identify insects in field, and just 1 to 3% of the insect collected each time are stored in absolute ethanol and then brought to the lab for the taxonomic identification to confirm the field identification, and then tested singly for Xylella fastidiosa by qPCR. During 4 sampling rounds of this monitoring program, instead, the monitoring is destructive. So we collect all the insects present in field and we tested them singly for Xylella fastidiosa. And we do so during like July, June, September, and October. So what has been identified as infectivity peaks of Xylella fastidiosa in olive orchard during like previous reports, previous works. But this like testing insects singly by qPCR is pretty time consuming and expensive. So in the frame of the Covexi project, we are testing an alternative approach. So we screen several plant species for suitability for both the bacterium and the spittle-bugs. And what we are proposing here is, instead of testing thousands of single vectors, like caging all the collected vectors on plants suitable for both the bacterium and the spittle-bugs, testing them for Xylella fastidiosa. So testing few plants rather than 1,000 insects. We can talk later about this. So agronomic management data are collected through questionnaires, which are administered to farmers. We ask like about the kind of management, organic versus conventional, data about the water management, fertilization, variety, but also the number of treatment they make, the chemicals that they use, and also if they implement biological control plans and the natural enemies that they release. And from this data, we are actually starting a project comparing the impact of organic versus conventional agriculture on Philaenis spumarius vector activity and on the transmission of Xylella fastidiosa. Several projects are stemming from this survey activity. And these questionnaires administered to farmers are part of also of a dissemination activity through which we like increase the awareness of people about what Regione Apulia is doing, like in terms of research, to try to counteract the spread of Xylella fastidiosa. So we increase the awareness in farmers, but we also organize activities with schools, stakeholders, private centers, which is pretty important in the territory. Additionally, so Philaenis spumarius is not just moving, like the active dispersal of Philaenis spumarius is pretty limited, as demonstrated by several papers, but one of the problem is the passive dispersal. So in addition to this like survey in olive orchard, we conduct also survey in parking areas, gas station, agricultural cooperatives, and additionally 90 sampling points north to the northernmost foci of Xylella fastidiosa ST53 that you can visualize on the map as these green triangles. So in these like sampling points, we collect on the vegetation, whatever the vegetation, all the insect vector that are then brought to the lab, identified and tested by qPCR for Xylella fastidiosa. And obviously, now it arrives the point why we are here today. is because of, in the frame of this activity in 2022, we detected in 2 olive orchards where olive plants are interspersed with almond and grape, 2 male of Xylella Seminars in 2022 that tested positive to Xylella fastidiosa by qPCR. And then in 2000, this in the municipality of Triggiano, which is actually few kilometers far from Bari, the capital of Regione Apulia. And in 2023, a kilometer from like the first detection site, we found additionally 13 individuals that tested positive to Xylella fastidiosa. And as Maria will explain, Maria conducted the multi-locus sequence type on this individual and confirmed that they were infected with Xylella fastidiosa ST1 strain. And obviously, after this finding, we decided together with the Regione Apulia to like implement this survey, including another activity, specifically in this outbreak of ST1 in Triggiano. So in addition to the sampling alongside the main roads and highways, and in addition to the sampling in olive orchard, we decided to like monitor 16 orchards in Triggiano, almond orchard or vineyard, to try to understand what vector species are involved in the spread of the ST1, what are the transmission pathways, and what are the plants that are involved in the outbreak of the ST1. So, and that's all for me. And please, the floor is Maria's.
Yes, thank you, Daniele, for introducing the topic, and to Evelina and Juan who introduced the seminar. Okay. So I will continue. Okay, I will continue with the activities that have been carried out on the plant and what. Which have been the implication of the finding that Daniele described. As he was saying, here in Apulia, we are unfortunately famous for the olive strain that causes the olive quick decline syndrome. And once the first insect, the first 2 insects we found in the municipality close to Bari, in the municipality of Triggiano, of course, the first action was to go there and to see if there were some symptoms. And the main target, or the main plant species that was targeted by the inspection was, of course, olive, because the hypothesis was that most likely we had a jump of the infection from the southern part to the northern part. For two consecutive times, olive samples were sampled along the site, close by the site where the positive insect were found, and all the trees tested negatives, nor the inspector could see any sign of a possible infection. So during 2013, we could not still answer to the question about the origin of this infected Phyllenus spumarius. In 2013, in late summer, early autumn, of course, this site was particularly prioritized for the monitoring, for monitoring the insect vector. When, as Daniele said, more infective specimens were found. At this time, this raised an alert in the area, and was the time when then in early 2014, a more in-depth inspection in the area was carried out. This time, more plant species were included in the sampling inspection. Of course, most of the species that you see listed here were in the dormant stage, so it was not possible to observe any symptoms, except, of course, on olives, on which again no symptoms were observed. For our surprise, from these 400 samples collected in the area at the end of January 2014, we got 6 almond trees positive to Xylella fastidiosa. So we worked this picture, the picture of these infected trees have been taken a few days ago. So at the time of the first identification, they were almost dormant or just sprouting. And so our question was why we are finding on almond, which generally is known to be not the preferable host of the ST53 strain, while we are not finding any infection on olives. So the hypothesis was immediately that most likely was not the strain spreading in the southern part of the region to be the causal agent of this infection. And in fact, as Daniele mentioned, we performed molecular analysis both on the plants, on these almond trees, as well as on the insect, and we discovered that the isolate occurring in these infected samples was not the pauca, but the fastidiosa, and specifically the sequence type 1. So based on the what were the implication of this finding? Based on the European legislation, when a new outbreak is discovered in a Xylella-free area, the first action is to demarcate the area surrounding the infected trees, and this was the case, and this buffer zone was delimited with a radius of 2.5 kilometers. Which are the action then in this buffer zone? Because one of the first requirements when we find a new outbreak is to define and to characterize the strain. Because we found the ST1, the plant species that are monitored and inspected in this buffer zone are those listed as susceptible to the subspecies to which the isolate and the strain belongs. So in the case of the subspecies fastidiosa, the European regulation lists 40 different plant species that, according to the EFSA database, have been reported as infected by subspecies fastidiosa. So in this area, it was started immediately a reinforced survey targeting 40 different plant species. And this is the situation of the results of this monitoring, the current, almost the current updated situation. The green dots are the trees that have been sampled and tested negative. The yellow one are those sampled, but the test is undergoing. And then you see the red one, which are unfortunately the numerous positive plants that have been found, which has been the monitoring strategy in this area. As I said, it was called reinforced monitoring, and it consisted each single hectare in this circumference has been inspected, and if in this hectare there were one or more of the 40 susceptible plant species, it was collected at least one sample for each species. Because of the high number of plants to be tested, as it is used in the pauca demarcated area, samples are tested by pooling more than one trees. specifically between 4 and 7. Currently, we are now doing in all cases 4 samples. And these are the results that our plant health services kindly shared with us, the latest data. In this, from the end of February, when the demarcation and the confirmation of the strain was obtained, in these past 2 months, more than 16,000 trees have been sampled, and 11,000 have been the test for 11,000 has been completed. And as you can see, the most the host plant that with the highest number of positive is almond, followed by grape. Whereas among the susceptible plants, susceptible species for the subspecies fastidiosa, there are also fig, citrus, peach, plum, and oleander. So far, none of these species has been found infected. So just to briefly introduce how the tests are performed, all the positive trees results from a double test, double checks that are performed in the Apulian laboratory, a first level test followed by a confirmation test that is performed in a second laboratory. At the moment, for the tree, for cherry, almond, and grapes, we have done the subspecies identification for all positives, except in the case of apricot, because the titer of this single plant that was infected is very low. So neither the real-time PCR using a specific primer for the different subspecies or the MLST analysis could be efficiently completed to determine the subspecies. And as I showed before in the slide before, the red spot, unfortunately, we're identified also. So positive plants were identified on the border of the buffer zone. This means that a new demarcation, the blue line that you see here, has been needed in order to delimitate a new buffer zone. So now the tests are ongoing in this area in order to assess how far and how big is this outbreak. Regarding the host plant found infected, here you see some almonds. As I said, when they were identified, they were at the early stage of the vegetative season, and currently they are in the full vegetation, and we cannot see any specific, as expected, for Xylella. At this stage, we cannot see any specific foliar symptom, but also we could not observe any symptoms of desiccation or decline on these plants. Because we found this outbreak in this period, which is not the optimal period for the testing and for detecting Xylella, which is known to be for which the best period is known to be in summer, late summer, and autumn. We are working. The detection is currently ongoing on xylem tissue, so scraped from the mature cuttings. And when we compare the detection on the leaves that we can collect now in spring with the xylem tissue, we can see a complete failure of the detection on the leaves, whereas the titer in the plants of the bacterium as estimated indirectly from the qPCR is in the range, the normal range of the qPCR results. As you can see here in this graph, the CQ value range from 24 to 32 with an average of 27. Of course, not all the cases of the trees of these more than 5,000 trees of almond, the results were, would say, crystal clear, so positive or negative. We still have a percentage of samples in which we assessed as undetermined, because the CQ values are above the threshold that generally in the lab as we are using, and I would say that at least at European level, these thresholds the threshold are quite debated, and it's difficult to have a threshold. But maybe the application of digital PCR can solve some of these cases in which we really don't know if the plant is positive, if the bacterium is multiplying in the plants or not. The same situation is for cherry, the other host that has been found infected. And the situation is similar for grapes, for which there were some report of possible bad failure that can be observed in the vineyard. These are example of infected plants in table grape in a vineyard with cultivar for table grape cultivars, but and in-depth inspections in this field did not confirm that these problems of sprouting in some of delay of sprouting in some of the plants are limited to the infected plants. What is interesting to say about the data that we are collecting, that the Plant Health Service is collecting, is that in this image on the left, it is shown all these rectangular light green color are table grape vineyard. So unfortunately, this outbreak has been identified close by an high intense agriculture area where table grape, one of the major producing area for table grape. So and these are the plastic that is used to cover the vineyard. And interesting, when in one of these orchards where at the beginning some grapevine were found, by testing the whole vineyard, only the infected plants are mainly restricted on the border of the vineyard. Similarly to almond, test is still on to be done on the mature vines, because if we compare the use of green shoots or leaf petioles compared to the xylem tissue, we have clear results of negative results in the young tissue developed this year, while we have a clear and clear detection in the vines. Even if at this period we can collect only small pieces of mature cuttings, mature shoots, because of course grapevine, the plants have been pruned last autumn. One of the challenges of this outbreak, speaking from the detection point of view, is that currently in the region we have now an area where pauca is widespread. Then we have this outbreak of fastidiosa, and unfortunately, a few weeks ago, it has been also discovered a multiplex outbreak also in the north of the region. So in all these outbreaks, the common host plant is almond, and based on what I told you before about the EU regulation, it is important to, when we find an almond tree is infected, to assess which is the subspecies causing the infection, because these affect then how the application of the monitoring program is developed. So if it's pauca, we know that the main host plants that should be targeted is olive. In the case of fastidiosa, we have the 40 species that I mentioned, and in the case of multiplex, other plant species. So when at the moment in the region, when we detect a positive almond, we need to provide to the plant health services, health service, also the strain of that is causing the infection. At the moment, the EU regulation include only the MLST analysis as the most widespread, commonly used approach. But this approach, of course, cannot be used for routine testing. So we could not test all the more than 100 infected plants that have been so far identified in the new outbreak. So we are now validating a test, specifically the one developed by the French colleague reported in DUPA et al. We are using the 3 probes specific to the 3 subspecies, and so all the positive samples goes to this flow of diagnosis, and we could assign the subspecies for the majority of the infected plants. Beside the molecular detection, of course, one of the initial one of the priority was also to isolate the bacterium. Again, we are working only on wood because of the season, and by ensuring a good homogenization, maceration of the tissue, we successfully isolated the bacterium from almond, from which we could recover a high number of CFU. The situation is different in grape and cherry. From this host, we could recover very few cells, if any, sometimes, while in almond this isolation succeed almost 100%. We are using different media, but those commonly used for fastidiosa, with the PWG promoting more growth than, for example, the PD3. We also tried to isolate by doing without maceration, but just directly from the imprints of the stem that we collect, and again we succeed to have the colonies, even if in a longer time compared to the previous method. And I conclude by thanking the team of Dr. Infantino from the Regional Plant Protection Service for sharing the data that I presented, but also for the huge efforts they are putting in the field in collaboration with the agency ARIF, which has the inspectors in the field, and all my team, in particular Donato, for some suggestion and the data I presented. Thank you.
Thank you, Maria. Thank you, Daniele. That was very, very full of information presentation. Thank you for that. Do we have a connection with Juan in the room? And some questions in the chat?